Hey, good morning. I hope you are doing good. And my name is Ramesh Krishnan. Today I'll be talking about an introduction to positional gene cloning in plants. I am working as an assistant project scientist at the Department of Plant Sciences at the University of California, Davis, for last 6 years. I did my undergraduation in plant sciences at NGM College Polachi, the same college who is organizing this FDP program. In the same college, I did my PG in biotechnology. Then I moved to Hyderabad for like 6 to 9 months, I guess. I think it's 8 months for my internship program at ICRISAT. There I worked on chickpeas, molecular breeding and some pathology, then disease resistance screening. Then I moved to Mysore for like 4 years for my PhD program. There I worked on mulberry molecular breeding. I developed DNA markers, some softwares, databases. We developed a core collection, screened like hundreds of germplasm using SSR molecular markers, if you know what SSRs are. Then I moved to UC Davis in 2014 for my postdoc at the Department of Plant Sciences. Here I'm working with Jan Dvořák at the Department of Plant Sciences. We mainly work on molecular breeding. We do genome sequencing of wheat and wheat ancestors, and walnut is a tree crop and it's a very important crop in California, USA. I do a lot of molecular breeding, genome sequencing, RNA sequencing, gene expression. Then we clone candidate genes. So the candidate genes are the genes who are controlling the expression of phenotypic traits in like important crops in California like wheat and walnut. So here is a link to my Google Scholar profile. If you go there, you can find out my publications if you're like interested. And also we put all these URLs under this YouTube video in the description, so you can go and click instead of typing all these. Don't need to gymnastics with this alphanumerics. Okay, this talk is not super basic talk. It's like if you don't have any knowledge on genetics, and probably you may feel it's hard to follow, and it's not super advanced though. So I want to go in the middle, like not super basic and not super advanced. If you have some undergraduate level or even like school level knowledge on plant breeding, like you will be fine. Whole idea about this candidate gene cloning using this way, like positional gene cloning, is mainly applicable to plants or the organisms those who can breed like animals like cow. For humans, it's not applicable. Some concepts in this talk are a little simplified, so I'm not going to talk about like cutting edge research happening in this field. I'm going to give you an idea about how people clone a gene. This video is slow paced. I want everyone in this group to follow this talk regardless of their background. So if you're not someone who is not used to this kind of slow videos, you can go ahead and click the gear icon of the YouTube and make it 2x faster. What is gene cloning actually means? Gene cloning in this context, it actually means how to isolate and characterize a particular gene of interest. Say like you are walking into your garden or your garden, and you are looking at a plant, say chickpea. So you have like few plants of chickpea, and few are like high yielding, and few are like low yielding. You want the high yielding plants, and you also want to know what are the genes actually controlling this yield. That trait what we are looking at is yield, that high yield or low yield. If you want to do that, then how you go about? Before I start talking about gene cloning, I want to go over some concepts in plant breeding and genetics. Say you have like two plants, plant 1 and plant 2. Let me use pen here. Yeah, you have a plant 1 and plant 2. Say this plant is high yielding and this plant is a low yielding plant. I think probably you guys know about chromosomes. What are the chromosomes are? If you don't know, like I give you a small introduction. So this is a plant cell. Like you have like trillions of plant cells. In the middle of that cell, you have a nucleus. In the nucleus, you have a DNA as a genetic material in plants. So imagine this DNA as a thread, something like this. Yeah. So the threads are arranged as a small bundles called chromosomes. So this number of chromosomes varies from species to species. So since we're talking about chickpea, my handwriting is so horrible with this tablet. Since we're talking about chickpea that n equal to 8 and 2n equal to 16. I will come back to why equal to why we are putting 2n. So for the rice, n equal to 12 and then 2n equal to 24. So we are putting 2n because it got one n from one parent, female parent, and other n from male parent. Let's say like this homologous chromosome from one parent, other from. So these two are called as homologous chromosomes. The H1 or H1 came from female parent, H2 came from male parent. Since you have like 8 pairs of chromosomes in chickpea, you get to see like one pair and two pair, and it keeps going for like till 8 pairs of chromosomes. One homologous chromosome came from female parent and other homologous chromosome came from male parent. So you may ask me why we are calling this homologous chromosomes. So gene in this homologous chromosome 1 here and the gene in the homologous here are same. One may be dominant gene and another may be a recessive gene. I will explain these things in the next slide, but they are all the same genes. Like the plant 1 and plant 2 all happens to have the same number of chromosomes because same species. So if you come here at the time of meiosis, one recombination even happens. Now imagine this is a one chromosome and this is another chromosome. So the crossover happens like this, and this piece of this chromosomes comes and joins with this, and this piece of this chromosomes comes and joins with this. So the crossover happens. This blue piece came from here to here, and gray piece came from here to here. So this blue piece jumped here and gray piece jumped here. This happens in all the chromosomes, like 8 chromosomes again here, since it's chickpea. At the time of gametic formation, here the 2n, you have 2 pieces. The only one piece goes to the gamete, like H1 and H1 or H1, H2 or H2, H1, whatever. So the 2n became n. Only the 8 forms the gamete, and this 8, and if you cross between these two, and this 8 and this 8 combines and form n, say like 8 plus 8 equal to 16. Then 2n equal to 16. That is this. Say, where is that? This one came from here, and from this piece, this one got here, and here this one, and with this. So the same event happens again and again. Like again, I'm not able to see the pointer here. So meiosis happens, again happens like this. This cycle keeps happening. When you read about some molecular biology papers or about cloning or genome sequencing, you probably read about this kind of jargons like DNA, gene, allele, locus, markers. What is DNA? You probably know that DNA. You have cell, I'm just repeating. You have nucleus and you have genetic material, that is DNA. DNA forms into a small bundles, those are chromosomes. Some species have 8, 16, 12, and length also varies from species to species. For example, I don't know about other crops. For walnut I'm working, that's about 650 million bases. So 650 million bases is something like 650 multiplied by 10 to the power of 6 bases. So you have like 80 GC repeats like for this many number of times. So now you ask me, this is DNA. What is gene? So gene also is a piece of DNA. So like here, but it has some functional unit. So genes are called the functional units of DNA. What it does usually, not all genes makes proteins. So if it makes proteins, DNA, the transcription happens. Say like again, I need to draw this. I need to go back to the nucleus. So inside nucleus you have DNA. Here transcription happens. It makes RNA. The RNA goes out and it forms protein, and protein goes and it does its job. So genes are nothing but functional unit of DNA. So you may ask me, what are these pieces are doing? We don't know yet. Some people call it a junk DNA. Some people say no, it is doing something, but we don't know it. And you may ask me then, what is an allele? Now you know about DNA, you know about gene. Then what is an allele? So allele is nothing but you have a pair of chromosome. The one came from female parent, other came from male parent. You have the same gene here. Let's say this gene. Let's call it gene 1. So this is called allele. So this is an allele of this and this is an allele of this. The other complementary piece of the gene is called an allele. So what is a locus? So you know like this is a gene, but you need to know the location. That's called locus. So how you say like this gene is somewhere, like you can say like this place is somewhere. You say the Karnataka is in India. It's a location, right? So how you say like what is the locus of this gene? I would probably say at the chromosome 1 at like 15th megabase, 15 point something like it goes megabase, you see this gene 1. So that's a location of the gene. So what are the markers? Now you know like DNA, gene, allele, locus and markers. You want to know the difference between this and this to differentiate. How would you differentiate? Say for example, this is your workplace. So this is your office, or you're teaching your college or university. This is your home. So now it's like 5 o'clock. You are now traveling from your work to home. You have multiple paths to take. Like you have this and you have this. You can take any road. Let's call road 1, road 2 and road 3. And from your home your mom is asking, hey, where are you? So what would you respond? You cannot say like I am but that 15th kilometer, like 15th kilometer from this. That's too difficult, right? So you probably in your real life you probably use called landmark. You probably say I am near some place, say like a village, or like what's the village? Village 1, village 2, right? Village 3. So you probably say if you're taking this path, you say I'm in village 1, village 2, village 3. And if you're taking this path, let's call this village village 4 and let's call this village village 5. You would say like I am at village 5. So your mom knows, okay, he is somewhere here. So why we are using these villages as a landmark? So you can pause this video and think. Because this village is not here. Imagine you have the same village, right? Village 1 is here, the same village is here. Then your mom will get confused. You're here or here, you don't know. So you need to have some unique place like village. It's like let's strike out. You need to have unique place like village 1 and village 4 here. Those things are called markers. If I'm comparing this homologous chromosomes with this homologous chromosomes, I need a difference. This base should be A and this base is T. Then you'll see the difference. Like if I start sequencing this A and this T, you know, okay, I am looking at, if it's A, I'm looking at this homologous chromosomes. Let's draw again H1 and H2. Here you have C and you have T. If I sequence this area, if I see T, then you know like, okay, this organism or this progeny has this homologous chromosomes, not this homologous chromosome from this parent. These markers are also highly useful to compare two different plants. Let's commonly called plant or genotypes. People say genotype because those have like different markers and these have different genomes. Let's say like here let's call plant 1 and plant 2. Say like you are comparing the plant 1 with plant 2, you need to have some difference between these two in the genome. And if you have like A and if you have T, if you sequence this area and if you get A, then you can say yes, this has this piece of DNA, like this genome, this chromosomes, not this allele. Say like this is A allele, this is called T allele. Now what is gene cloning? So you know that gene cloning is isolating and characterizing a gene of interest. Usually the gene controlling our trait of interest, some phenotypic trait of interest, like high yield. There are multiple ways to clone a gene. Famous one is reverse genetics. Yield is a very highly complex trait. High yield may happen due to expression of multiple genes. Say I am interested in a color of flower. This one is in rose. It's a rose color, and this is in red. So this probably due to the pigment variation between rose and red. If you know that what is the pigment is controlling this color from that amino acid sequence, you would be able to tell what is an RNA sequence going reverse. Then you can tell what is a DNA sequence. Then you can say what is a gene sequence. Other way is comparative genomics. Say like rice and wheat are related. If I clone a gene for a high yielding in rice, the same gene may contribute to the high yield in the wheat. If I have the sequence, genetic sequence or gene sequence for rice, I may go and look at that wheat genome. If I see the same sequence, I probably knock the gene out and look for the change in phenotypic trait. If I knock out the particular gene, then I see that there's a change in yield, then I would say yes, the same gene is controlling rice and wheat. So if you don't have any of this information, that's a case usually. So some traits, like most of the traits, we have no information of any of these things. Most of the traits are like, it's not very easy, something like rose or red. Those are like yield. We need to use this forward genetics. So here are the like couple of papers. These are like really useful papers. Like we put all these URLs in that YouTube video description, so you will be able to go ahead and read all this literature. So I'm going to talk mostly on this forward genetics thing. Here is a schematic diagram from this paper. You can again, we will put this link in the description. You can go ahead and read this paper. This is a recent paper in 2019 or 20 from my old boss, Dr. Rajiv Vaishnav from ICRISAT. Sequencing is getting cheaper and cheaper. In the pre-NGS era, you usually develop mapping population. So mapping population is nothing but you have again high yielding plant and you have low yielding plant. If you cross these two plants, you get F1. And if you again cross F1 with F1, you get F2. So again, I will explain about this cross in future slides. Once you have that plant breeding population, you go ahead and phenotype and look for this is a high yielding or low yielding. You plant them in the field or in the greenhouse and look for the trait phenotypic variation. At the same time, you do genotyping like RFLP, RAPD, AFLP, SSR, SNPs are very popular. What is genetic map? You sequenced a genome, or you know the markers of a piece of DNA. Only nowadays, because of sequencing is cheaper, you have to sequence the whole genome. So usually what people do is they only sequence small pieces like this. Yes, it's cheaper, right, to sequence a small piece instead of whole genome. Still, genome sequencing cost a lot of money, like thousands and thousands of dollars, but it's cheaper compared to like 8 or 9 years back. So you have like a low yielding plant and you have high yielding plant. We need to do the genetic mapping. We need to place these markers on the chromosomes. So how would you place these markers on the chromosomes? It's very simple, right? There is a simple law. It states that the distance between the markers is directly proportional to the higher number of recombinations. If it is a small distance, you have very less recombination. Imagine I am giving you a thread. On the thread, I am writing A at here and B here. The length of the thread is, let's say, 15 centimeters. I am giving you a scissor and asking you to cut at any place. So what's the probability of cutting between A and B? It's almost like 100%. If not 100, it's probably like 99.99% probability that you cut between these, because it's sitting at the very end. Say like I'm giving you a thread. I'm placing this A here at the same very close B. I'm again asking you to cut the thread. So what's the probability of the cut between A and B? If not 0, you said probably between 0.0000 some 1 or something, like the probability of cutting. Because they are very close, they are next to each other, you cannot cut. The probability is so less. Same concept applies here. If the markers are far apart, the probability is higher. The markers are very close by, the probability is very low. So based on that, you would be able to place the marker on the chromosomes. Say if I have 50 markers, there's a lot of recombination happen on all this, like here it's recombination happen, here recombination happen, here recombination happen, here, here, in the here, here you have a lot of recombinations. Then I count the number of recombination between these two markers, 2. Then I say, what's the recombination between these two? I say 1. And then I count, what's the recombination between these two? Let's say 5. And say, what's the recombination between these two? Then 6. And I'm going to ask, then what's the recombination even between this and this? You say 5, 6 plus 2, 8. So by doing this, you would be able to order the molecular markers on the chromosomes. Like by doing this, you'll be able to order like this. This is a marker 1 and marker 2 and marker 3. So now we ordered all the markers on this chromosome. At the same time, you also know what's it number of recombination between this 2. It's a 5. Then you say marker 3 and 4. These are quite far apart because you have 5 recombination compared to, so these are very close, right? But here this is very far apart because you have 5 recombinations even between this. So by doing this, you would be able to map the whole genome, and you see the number of recombination events happen between these two, and you ordered all the markers on all the chromosomes. Say chickpea, and you have like 8, like 1, 2, 3, 4, 5, 6, and for all the 8, remember, for all the 8 chromosomes, you have the complementary homologous chromosome. We did the genetic mapping. Now we need to know what is a QTL mapping. So first we need to understand what is QTL. QTL means quantitative. I'm not going to write. Yeah. So QTL means quantitative trait locus. So you know what is quantitative. It's a plain English. And you know the trait. Trait is nothing but a phenotype. Here, high yielding, low yielding. You know, locus. Locus is what? Locus is nothing but location on the chromosome. Location. This is a trait. We call it a locus. So we need to map where is that location. Here we are not going to map the gene, but we are going to map on what place of the DNA or chromosome is controlling the trait. Again, I'm going to draw the same example here. We have the homologous chromosome 1 and homologous chromosome 2. Again, homologous chromosome 1, homologous chromosome 2. The plant 1 is again high yielding. Let's put high. Plant 2 is again low yielding. Let's put 2. And this is a gene. Gene. This is a gene. So these two genes are allele, right? This is a high yielding gene and this is a low yielding gene. But that both the genes are doing same job. That's why it's called allele, and these are on the same locus. And you are having F2. It's getting one chromosome from one parent and other chromosome from other parent. So what again happens here? Again it happens so many recombinations, because you are not looking at one plant, right? You are looking at multiple plants, hundreds of plants. Usually we look at like 300 to 500 plants. So you look at like 500 to 600 recombinations. Say like you are looking at 500 plants, you are looking at 1000 recombinations, right? 500 from one parent and 500 from other parent. Now we have F2 progenies. So the F2 breeds with itself. So we cross F1 with F1 and the same recombination even happens. The same recombination happens and it gets one chromosome from here, another chromosome from here. So you get one homologous chromosome from here and homologous chromosome. So this is your black box. So black box is your gene. black box is your gene. What happens here is some plants get, let's put this is an high yielding gene, let's put Hg is a low yielding gene, Lg. Some plants get high yielding genes, what's called Hg, and some plants get low yielding genes, Lg. Based on these markers, now we know the blue markers, right? Those are the known landmarks, but you don't know about this piece of DNA where the gene is sitting on. Since you know these markers around the gene, you would be able to tell, hey, here you have green, here you have blue, so probably this, so recombination even happened here, probably the gene is here. Again, here it's a green, here it's a blue, it's here it's a green, here it's a blue, its gene is here. See, based on this recombination event, you will be able to tell the location of the gene. So based on the recombination events, let me draw again, like say, let's see, here you have high yielding gene and here you have low yielding gene. If the recombination event happens here and this thing goes out, instead you get this thing, the plant will become a high yielding plant. Again, if the recombination event happens like this, and if you get a high yielding gene, the plant will get a high yield. If it gets low yielding gene, the plant will become low yield. So based on this information, you will get to know the location of the gene on the chromosome, right? Here I am giving an one example. I just copied from this Nature paper from 2001. This is an amazing paper. I think you go ahead and read this paper if you guys are interested in plant breeding or this molecular breeding stuff. Here you have one parent, parent 1, and you have other parent, parent 2. Here you have like this hairy structure, you have a large number of hairy structures. Here you have a very few. If you cross the F1, right, like you are crossing this, and it's getting one from here and one homologous chromosome from here, and you are having somewhere in the middle, right? And you are crossing with F1 again and again, and so you are keep doing this till F8. So like F1 is like you are crossing F1 and F2, you will end up with F2. So again you cross with F2, you will end up with F3. Again you cross with itself, F3. You go with F2. So you keep doing because you will get more recombinations on this chromosome. Say it's like chromosome. If you have like crossing like 8 times F equal to 8, then you are going to have like 8 different breaks. More the break, you get more the resolution, right? So usually people for some traits that F2 is enough. For some traits people go till like F6 or F8. The only issue with F8 going till F8 is it's gonna take very long time. Like every year, if it's a seasonal crop, you can just cross only once. Say like walnut, like if you cross it, like creating F8 would take like 20 to 40 years. That's a long time because it's a tree crop, right? For some crop, it's like tomato, it's probably like quicker. For some crops like tree crops, no, it's not gonna happen because it's take 40 years. In a real life, you don't get that much resolution. So you say this is a chromosome, like say this is a chromosome. You will end up like from here to here. I think from somewhere in this region that gene is sitting. The QTL is such a long. Sometimes even if you end up with, once I ended up with like 12 megabases in walnut. Under 12 megabases, I got like 800 genes. If I'm not wrong, I think 600 to 800 genes I got. So can you go and check the 600 to 800 genes in walnut? No way, it's not possible. Again, it's a tree crop, and even in like rice, it's not possible to go and check like each and every gene in that interval to see like is this gene related to that particular trait or not. No way, this is not going to happen. So what people usually do is, again they go back and cross a population and they select only the plants that progeny is having recombination between this to this area, right? So if you have like a lot of breaks like this will be really helpful to understand where the gene is sitting. 2 years back when I was working with walnut, first I ended up with like 8 megabases interval. Then we crossed that plant again and again and again and again. Then we only selected that recombination even between that 8 mega, and we ended up with like 1.6 megabases. The one even 1.6 is quite big. Like the 1.6, if you imagine like 1 to the 1.6 10 to the power of 6 bases, like, and ended up with genes. So then I checked like individually like 60 genes. So what are those genes? Here you can see that what are those genes under in that location. They're probably like contributing to that trait. So once you have the gene, then the life gets very very easier. So once you have the gene, so how do you know that gene is actually controlling the particular trait, right? You need to validate and you need to cross check. So again, I will go for gene expression. So I have a drawing plant 1 and plant 2. This is high yielding and this is low yielding, right? Here I will look at that, say like I shortlisted instead of one gene, I shortlisted 4 genes. Gene 1, gene 2, gene 3, gene 4. I look at the gene expression of that particular gene in this crop, also in this plant. Let's put it this way: gene 1, gene 2, gene 3 and gene 4. If I see the higher expression here like this, I'm just putting it line graph. If I see the higher expression here, gene 1, but here I don't have no expression, no expression, no expression. Here no expression, here no nothing, very less expression. Here there is no difference at all. So we can say this gene is not related, and you will be able to say yes, there is a difference in gene expression. So like here I'm getting like, I'm just giving like 600 copies of the gene. It's not the right way to express anyway. So for example, like I'm getting 600 copies of RNA, and here I'm getting only like 50 copies of RNA. So like you can say like yes, this gene is doing something and it's controlling this particular trait. Okay, now you have the step 1 validation. Okay, step 1 validation. Let's put step. So what you do next is gene knockout. You go to this high yielding plant and remove the particular gene from the genome, and you see the expression. Again, let me draw here. Oops, I'm not able to draw straightly with this tablet. So this is a high yielding plant and this is a low yielding plant. I am going and removing this gene 1 here, just striking out. I am keeping this gene here, and you should able to see this plant should turn low yield, and this always stays at low yield though. So it means by knocking out I mean removing the gene, you would be able to tell yes, this gene is controlling a particular trait or not. I think that I should stop here. So I hope I covered that basics of genetics. Then I moved to gene cloning. I hope I explained about how the gene cloning works and what is the methodology. And if you have any questions, or like if you think you have some question, you can always email me. Here is my email address. And thanks for your time and thanks for listening. And I wish you all the best. Stay safe and stay healthy. Thank you. Bye.